Welcoming our next speaker, Dr. Jelen, Senior Registrar in Psychiatry at the Maudsley Hospital, who very recently, I may add, completed his PhD in neuroimaging, supported by the MRC Clinical Research Training Fellowship. And he's going to talk to us about the GOMARK study, glutamate and opioid mechanism of antidepressant response to ketamine. Thank you.
Perfect, so much. Can everyone hear me at the back? So, thank you very much for the introduction and for the invitation to present some of my preliminary findings from my PhD investigating glutamate and opioid mechanisms of antidepressant response to ketamine. So, apart from the generous funding from the Medical Research Council, no other conflicts of interest to declare. So, the exact neural mechanisms underlying the antidepressant effects are yet to be fully determined. This is what we're here trying to figure out. Work to understand ketamine's mechanism of action could ultimately lead to more targeted and effective depression treatments in the future. One view suggests ketamine preferentially blocks NMDA receptors expressed on GABAergic interneurons, leading to disinhibition of pyramidal neurons and increased glutamate release. This so-called glutamate surge stimulates postsynaptic AMPA receptors, leading to activation of neuroplastic signaling pathways and synaptogenesis, which downstream may ultimately lead to brain network reorganization. Now, although ketamine is primarily described as an uncompetitive NMDA receptor antagonist, it also interacts with a range of other receptor systems, including GABA, GABAergic, dopamine, serotonin, cholinergic, and as we've just heard, opioid. Focusing on the interactions with the opioid system, ketamine has micromolar affinity for mu and kappa opioid receptors, produces opioid receptor-dependent analgesia, potentiates opioid analgesic effects, and at much higher doses produces mu-opioid receptor-dependent respiratory depression. However, less is known about the role of the opioid system for the lower doses used for ketamine's antidepressant effects. We know ketamine has known abuse liability and produces reinforcement and antidepressant-like effects in rodents. Preclinical work has demonstrated that activation of opioid receptors is necessary but not sufficient for ketamine to reduce depressive-like behavioral symptoms, suggesting more of a permissive role. Further preclinical work that Marjorie introduced has suggested that the abuse liability of ketamine is mediated primarily by its S enantiomer and involves direct interactions with brain mu-opioid receptors at doses and concentrations relevant to its reinforcement and antidepressant-like efficacy. Now, looking at the clinical evidence, in a pivotal study by Nolan Williams and Professor Alan Schatzberg, in 12 subjects with treatment-resistant depression, it was found that pre-treatment with naltrexone, an opioid receptor antagonist, significantly attenuated antidepressant and anti-suicidal effects of ketamine. However, this was a relatively small study, and other work in individuals with comorbid alcohol use disorder failed to replicate this effect. The conclusion from the Williams study was that larger clinical replication studies were needed to clarify the role of the opioid system in ketamine's antidepressant effects, but also to further explore potential mechanisms that may underlie these effects. So this is exactly what we sought out to do in the GOMARK study. First, as a clinical replication of the Williams study, however, with a mechanistic focus by investigating the acute effects of ketamine and opioid receptor antagonism on neuroimaging measures as well as the clinical outcome measures. So we conducted a randomized, double-blind crossover study comprising two treatment conditions, oral placebo or oral naltrexone at a 50 milligram dose preceding ketamine infusion, given at 0.5 milligrams per kilogram over 40 minutes. But this was administered during magnetic resonance imaging. And this was we had 26 completers, adults with major depressive disorder, and they were all within a major depressive episode. Depressive symptoms were rated immediately prior to each infusion and at post-infusion day 1 using the clinician-rated Montgomery-Åsberg Depression Rating Scale and the self-report QIDS. We also assessed anhedonia specifically using the self-reported SHAPS and TEPS measures. There was a washout period of 2 to 4 weeks between each dosing session. So here's a summary of the overall neuroimaging protocol. Participants were in the scanner for a total of 90 minutes. We first collected structural scans before a baseline resting state and arterial spin labeling scan. Next, we collected magnetic resonance spectroscopy data for a 5-minute baseline period, and then continuously through the first 30 minutes of the 40-minute ketamine infusion. In the final 10 minutes of the ketamine infusion, we collected a repeat arterial spin labeling scan, and this is to measure cerebral blood flow. And as soon as the ketamine infusion finished, we collected a final resting state scan. Today, I'm going to be focusing on the spectroscopy and arterial spin labeling findings. So magnetic resonance spectroscopy is a neuroimaging technique that allows for the non-invasive measurement of a wide range of neurometabolites, including glutamate. Now, a significant limitation to standard magnetic resonance spectroscopy is that measurements are determined over a single static time period with a kind of single average, preventing an understanding of potential dynamic neurochemical systems. In this study, we utilized functional magnetic resonance spectroscopy, where spectra acquired across time to investigate dynamic changes in brain glutamatergic metabolites. As I said before, we started by acquiring spectra continuously from an anterior cingulate cortex region of interest voxel for this 5-minute baseline period, and then during the first 30 minutes of the ketamine infusion. This is a bit of a summary of our analysis pipeline for the MRS data, pre-processed using something called the FIDE toolbox. Spectra were averaged per subject into 7 blocks. First, the baseline block, and then the 6 infusion blocks. These were analyzed using LC model. We had to exclude data from 2 participants due to poor MRS quality and some missing data. We determined changes in GLX, so the combined measure of glutamate and glutamine. This is commonly done at the 3 Tesla field strength we use. And this was referenced to total N-acetyl aspartate for each infusion block. And we compared that to the baseline to look at the change in GLX. And used linear mixed effects models to determine the effects of pretreatment condition, that's placebo or naltrexone, and then infusion block on the GLX change from baseline. So looking at our participant demographics, we had a relatively young sample with a mean age of 35 and an equal proportion of males and females. Participants had failed to respond to at least 2 antidepressant therapies and were experiencing moderate to severe depressive symptoms at the screening visit. Here we have some of our clinical measures, first of all. So these are the MADRS results presented here. So this is the MADRS plotted for pre-infusion at day 1 post-infusion. We have the placebo pre-treatment in blue, the naltrexone in orange. So the first thing to notice, very clear main effect of ketamine with reductions in mean MADRS scores at day 1 post-infusion. For the placebo group specifically, the mean scores reduced by 14.65 points, and for the naltrexone, this was 10.5, and we had a significantly attenuated reduction for the naltrexone plus ketamine condition, as demonstrated by a significant condition by time interaction. For the self-report QIDS and anhedonia-specific measures, although there's a significant main effect of time with day 1 reductions for both placebo and naltrexone groups, although the reductions were numerically lower for the naltrexone group, there were no significant effects of condition or condition by time interactions. Here we have the results for some of our subjective measure outcomes that included the CAD scale on the left, and then something called the psychotomimetic states inventory on the right that's split into various subscales. These measures were administered immediately after the ketamine infusion, with participants asked to recall the peak drug effects while in the scanner. And as you can see here, absolutely there are no significant differences in any of the subjective measure scores between placebo and naltrexone pretreatment. Next, moving on to our primary outcome, examining glutamatergic measures. Here you can see the change in GLX across the 6 infusion blocks, for placebo pretreatment condition again in blue, and for naltrexone in orange. Now, although it looks like there may have been an interaction here, in our model, the effective condition was only significant as a main effect, with a higher mean increase in GLX during ketamine for placebo compared to the naltrexone condition, suggesting that in our sample, opioid receptor antagonism was attenuating ketamine-induced glutamatergic activity in this anterior cingulate cortex region of interest. Next, in our study, we employed 3D pseudo-continuous arterial spin labeling scans to assess cerebral blood flow changes due to ketamine and naltrexone pretreatment. Our analysis focused on two main areas. First of all, a region of interest analysis, and then we looked at a whole brain voxel-wise analysis. And through these methods, the aim was to investigate how ketamine and naltrexone pretreatment influence cerebral blood flow across different brain regions. So first, we have the results from our region of interest analysis. First, on the left here, we have the pre-ketamine and global grey matter cerebral blood flow values during ketamine administration. And then the same for each of our 9 regions of interest on the right. Again, we had placebo pretreatment in blue, naltrexone pretreatment in orange. Now, we saw a similar pattern of results for global changes in cerebral blood flow and across each of our 9 specified regions of interest. There was a significant main effect of ketamine and pretreatment condition for all 9 regions of interest. What this means is that cerebral blood flow increased during the ketamine infusion compared to the pre-infusion scan, and there was a higher mean cerebral blood flow across scans for the naltrexone condition compared to the placebo condition for each ROI. We saw no significant interactions between ketamine and pretreatment condition for any of the regions of interest. The largest increases in cerebral blood flow we saw were observed in the pregenual and subgenual anterior cingulate cortex ROIs during the ketamine infusion. Next, moving on to the whole brain voxel-wise analysis, this revealed that ketamine significantly increased cerebral blood flow in a large frontal cluster encompassing the subgenual, pregenual, and dorsal anterior cingulate alongside the anterior prefrontal cortex and bilateral insula. We did not observe any significant pretreatment condition or ketamine by pretreatment condition effects in the whole brain voxel-wise analysis looking at cerebral blood flow changes. Now, this main effect of ketamine finding nicely mirrors previous work from our department examining acute cerebral blood flow changes during ketamine administration in healthy volunteers. Now, looking at some correlations with clinical measured changes, we found that for the placebo pretreatment condition, lower baseline relative cerebral blood flow in the subgenual anterior cingulate cortex, specifically Brodmann area 25, predicted greater antidepressant response to ketamine at day 1 post-infusion. Here, this was for the MADRS. Does this spot here? Here we are. Oop. Why is this not working? Anyway, MADRS and then the quids also. For the subjective measures, we observed significant positive correlations between the cerebral blood flow increase in the pregenual anterior cingulate cortex region of interest during the ketamine infusion and the delusional and perceptual distortion items of the psychotomimetic states inventory. Again, only for the placebo pretreatment condition. These associations were again abolished by the naltrexone pretreatment. Next, as an exploratory analysis for the arterial spin labeling data, we examined how changes in the spatial distribution of cerebral blood flow correlated with receptor density profiles, aiming to uncover the potential influence of different neurotransmitter systems to the cerebral blood flow signal. So it's a bit of a complicated figure here. I'll walk you through it. So we first generated unthresholded contrast images from our cerebral blood flow maps for 2 separate conditions. First of all, we focused on the placebo pretreatment arm and compared ketamine to pre-ketamine to almost get a main effect of ketamine without the influence of naltrexone. And then secondly, we looked at the interaction effect of ketamine by naltrexone treatment. And each of these contrast images was divided according to a parcellation atlas. We then obtained volumetric PET images that are indicative of receptor densities from public sources and parcellated these using the same atlas. Our focus was on mapping the densities of mu-opioid, kappa-opioid, glutamatergic, and GABAergic receptors, aligning with our hypotheses on interactions among these neurotransmitter systems. And finally, we applied Spearman correlations to assess the spatial relationship between the changes in cerebral blood flow, contrast profiles, and each of these receptor density profiles. One issue we face in running these sorts of correlation with brain imaging data is that of spatial autocorrelation. So to account for this, we generated 10,000 spatial autocorrelation preserving nulls to adjust our significance accordingly. So I'm not sure how well this projects with the light, but basically our spatial correlation analyses revealed a moderate correlation between the ketamine-induced changes in cerebral blood flow and the mu-opioid receptor distribution that remained statistically significant after the correction for spatial autocorrelation. We also examined further positive correlations between the ketamine-induced changes in cerebral blood flow and mGluR5 and GABA-A alpha 5 receptor density distributions that also remained significant after correction for spatial autocorrelation. Next, when we looked at the ketamine by naltrexone interaction for the cerebral blood flow contrast map, there were moderate correlations again with the mu-opioid receptor and GABA-A alpha 5 receptor density profiles that were again significant after spatial autocorrelation adjustment. So, attempting to bring some of these findings together, how might opioid receptor antagonism attenuate ketamine-induced glutamatergic activity? Now, we talked about the disinhibition hypothesis already today, which suggests ketamine selectively inhibits NMDA receptors on GABAergic interneurons, resulting in the disinhibition of pyramidal neurons, increasing neuronal activity and triggering the so-called glutamate surge. Now, importantly, mu-opioid receptors co-localize with NMDA receptors that are also located on GABAergic interneurons, including somatostatin and parvalbumin interneurons. Now, mu-opioid receptor activation normally acts to further reduce GABAergic inhibition and can enhance glutamatergic signaling. So this suggests that ketamine's potential dual role as a direct or indirect mu-opioid receptor agonist, possibly through beta-endorphin release, and NMDA antagonist effects on the GABAergic interneurons could synergistically disinhibit pyramidal neurons, leading to a significant increase in glutamate release. Now, we found a significant relationship between the spatial distribution of the naltrexone interaction effect on the cerebral blood flow changes following ketamine and the distribution of mu-opioid receptors and these GABA-A alpha 5 receptors. The GABA-A alpha 5 receptors are localized quite specifically on distal dendritic regions on pyramidal neurons that are principally targeted by the somatostatin interneurons, where they regulate tonic inhibition. So considering the localization of GABA-A alpha 5 receptors, our findings tentatively suggest, in the presence of a naltrexone, via the mu-opioid receptor antagonist effects on these GABAergic interneurons, an increase in GABAergic inhibition of the pyramidal cells could lead to a reduction in ketamine-induced increases in glutamatergic activity. So just to summarize what we've talked through, so in our sample of 26 adults with major depressive disorder, naltrexone pretreatment attenuated clinician-rated antidepressant effects of ketamine at day 1 post-infusion. Naltrexone pretreatment did not specifically attenuate anti-anhedonic effects of ketamine at day 1 post-infusion. Naltrexone pretreatment attenuated ketamine-induced glutamatergic activity. Naltrexone pretreatment did not significantly alter ketamine-induced focal cerebral blood flow increases. The spatial patterns of ketamine-induced cerebral blood flow changes and naltrexone pretreatment correlated with the distribution of mu-opioid, glutamatergic, and GABAergic systems. So, to conclude, opioid system activation may contribute to ketamine's acute antidepressant effects. Naltrexone may lessen ketamine's efficacy by attenuating glutamatergic activity. Our study offers insights into opioidergic and non-opioidergic mechanisms underlying ketamine effects and highlights the need for further research on the interaction between glutamatergic and opioidergic systems in ketamine and other antidepressants' therapeutic effects. So finally, to acknowledge excellent supervisors and input from physics and imaging experts, to all the amazing clinical research facility staff who supported the patients and me through this pretty challenging study, and finally to all our courageous study volunteers. Thank you.
Thank you, Luke. Any question? David, for that? Many questions. Wait.
Thank you, Luke. I know I got to overhear you during this last break talking about the kinds of experiences that the subjects would have in the scanners and how intense those experiences would be. And I'm curious if you noticed at all, maybe this wasn't formally assessed, I apologize if I missed this, if there seemed to be a different emotional valence to the experience or different kind of subjective quality between the groups?
So I remained, blinded obviously until the end of the study, and that was obviously, all the at the front of my mind, as I was sat and discussing the experiences with participants. There were some participants that very clearly described, one of their dosing sessions being quite positive, full of love, and the other was like much more darker and negative and what have you. And that probably was the main factor that influenced when we assessed blinding. It was the kind of quality of the experience or maybe side effects that influenced our guesses. However, when the unblinding came, it didn't necessarily match up like that at all. It really, so we actually did assess blinding right at the end of the study on the final visit, and that was, upheld.
Is that data published as well, the blinding?
It will be.
It will be, okay. Great, thank you.
Do you have that note of the guess? I mean, how did the guess line up with the outcome?
The guess we unfortunately only did at the end of the final dosing session at 24 hours. So we only have, one measure. We ran that, both the investigator and the participant guess, to see if it influenced the MADRS result, and it remained significant.
All right, thank you. Oh, Ami.
Yeah, hi. Yeah, so I think you said in the beginning regarding the first study that was published from Stanford that there was not enough power, it was only a small study, but your N is also the same, and I understand these imaging studies are difficult to do, but what was your primary variable? Did you have power to do any of the analysis?
Yes, so we had 26 completing the crossover. In the original Stanford study, they had 12 completing the crossover. So we did the power calculation based on the effect size they saw in their clinical measure, which was 0.7 when you included their whole treatment arm. But with our spectroscopy measure, especially with the repeated measures, we were powered with the 24.
For all the imaging variables too?
Not necessarily all of them, especially when I didn't present the resting state, but with resting state with signal dropout and movement, our total completer sample with adequate data dropped down to 20. Thanks.
Over there, Dr. Schatzberg.
Yeah, without crossing the people over, we had 14, and the effect size is virtually the same. We had 0.7, you had 0.6 for naltrexone. So, and we had no effect on CADS with naltrexone, and you had no effect also on CADS. So it doesn't change the dissociation; it changed that immediate antidepressant effect. What I think Luke didn't present, and what we we had an a priori that we would start with ketamine plus placebo responders because we could identify that pre-treating that they were responders. And we asked, do you block that effect in those people? And then it's 100% blocked. So you get a lot of softening because you're including ketamine partial or non-responders with even placebo. So but there's enough power if it's set up as a proper crossover. You don't have to have. Zarate only had 17 people when he did his study in 06.
Thank you. We have some question on the other side, and then I'll come back to Dr. Javed. Sivan over there.
Thanks. Brilliant talk, by the way. It was really nice to see some fMRS there. But my question for you is, I wanted to know what magnetic field strength you used, and also because if it is, for example, 3T, with the GLX, were you able to do some analysis to try and separate the glutamate and the glutamine? And also, did you manage to do any analysis on GABA? I'm just curious to maybe some time course analysis.
Yeah, perfect. So our study was at 3 Tesla. We used a press sequence, so specifically focused on glutamate rather than GABA. Yeah, with hindsight, it would have been beautiful to have mega press and look at both GABA and glutamate. We didn't at 3 Tesla, it's a bit unreliable to separate them, but I obviously did run those analyses just as exploratory, and it looks like the effect is driven more by glutamine than glutamate.
That's really interesting. Thank you for letting me know that. Thanks.
I think we have a question over there.
Yeah, thank you for this great talk. Just a quick question. For the studies, were the participants did they listen to any kind of music or have eye shades, or were they just in the fMRI or in the MR?
Yeah, so this was a point of discussion right at the very beginning of the study, because I was highly anxious about putting this vulnerable patient group, for most of them experiencing ketamine for the first time in the scanner. So I was thinking of any methods we could do to kind of make it more pleasant for them, including music and what have you. Following the discussions with the supervisors, they felt music would just be another extra variable influencing whether they like the music or don't like the music. So we chose to leave it out. I have to say I was surprised with actually how well tolerated it was in the end. There was a lot of preparation at the screening visit, carefully walking people. They had an opportunity in the mock scanner, trying to talk about what they might experience and what have you. And I think some of the kind of dissociative qualities, some of them even forgot they were in the scanner, forgot they were in the study, which seems, comparing the doses I've administered in our clinic, for example, at the same 0.5 milligram dose, there seemed to be something about that scanner environment that kind of upped the intensity.
Thanks. I think we have time for one last question, Dr. Javed, over here. And while Alicia is walking by, any difference in terms of people who were taking any other antidepressant? I saw some of your sample were taking some antidepressants, some were not. So
we had about half of our sample were currently taking antidepressants. Yeah. We kind of maintained them throughout the studies. Because our sample is the lower end of treatment resistance compared with the original study from Stanford.
So naltrexone has a pretty profound first-pass effect in the liver and sort of a variable area under the curve on pharmacokinetics, whereas naloxone is pretty much 100% inhibition. Has anybody tried this with naloxone nasal spray?
Not to my knowledge. The reason we stuck with the oral 50 milligrams was we had the kind of first study to kind of base this on, so we didn't want to kind of introduce new potential variables. But I think that's the way to do it. Also, naltrexone's not specific for the mu versus kappa opioid receptors, so further work when we're trying to probe this and narrow it down, we need more selective opioid antagonists.
Fantastic. Thank you. Thank you
Thank
very
you so
much, Luke.